Welcome everybody. My name is David Wood and I chair London Futurists. London Futurists is for people who want to think seriously about how the future might be significantly different from the present. It's for people who want to think both creatively and critically about possible future scenarios. One topic that has often featured in our meetings is the subject of the future of health, the future of healthcare, the future of medicine, and the future of aging. Such ideas have been the main topic of perhaps 3 or 4 of our meetings every year since the very first meeting I chaired of London Futurists 14 years ago in 2008. We're returning to that same topic today, but not to simply repeat what's already been covered in these previous events. Instead, today's speaker is going to take a notably different viewpoint on several key issues about the future of aging. Today's speaker is Dr. Jean Hébert, who is professor at the Department of Neuroscience and a professor in the Department of Genetics at the Albert Einstein College of Medicine in New York. Jean also heads the Hébert Lab at the Albert Einstein College, and that lab is dedicated to moving towards restoration of brain function with tissue engineering. Jean previously held academic positions at Stanford. He earned his PhD in genetics from the University of California, San Francisco, and he studied as an undergraduate in McGill University in Montreal. Jean, many thanks for joining us today. Over to you to set the opening stage.
Thanks so much for having me, David. It's an honor to be on your webinar. And yeah, so I'll be discussing what one aspect of the future that I think will inevitably happen. That's beating aging, but I think it's going to happen in a way that most people are not thinking about it today. And so I'd like to share that with you and get your thoughts in return. So I have some slides that might be useful just to get everybody thinking about this. So I'm going to go ahead and share that. Okay. I'm not going to, you guys can see the slide okay? It's not too small or, okay, good.
That's good, thanks.
Good, good, good. So the way I think that we're going to beat aging is by replacing cells, tissues, organs, even some more wholesale approaches to replacements. And that is even for the brain. And that might sound odd at first because the brain largely defines who we are as individuals. But there are good reasons for thinking that that is doable as well, at least progressively over time. So a lot of us think that health is important and that aging is a big problem when it comes to health. It causes lots of diseases that kill us. And even in the absence of disease, it makes us frail over time, even if we're perfectly healthy 95-year-old, our life expectancy is very short, even in the absence of disease. So the best way to live longer and healthier lives, I think everybody agrees with this, is to fight or eliminate aging. So, you know, I think it's important. Aging, everybody in the longevity field has their own definition based on what they look at. But I think aging was best described decades ago by biochemists who looked carefully at what happens to all our tissues when we age. And it was pretty ugly and sort of turned them off of the topic. So they kind of abandoned their hope that we could ever fight aging because the amounts of damage that occurs, not at the cellular level like some talk about, that's not really damage. Those things are performance of cells. But at the macromolecular level, so the material that we're made of is proteins, DNA, carbohydrates, and lipids. And they all accumulate these complex forms of stochastic damage, so chemical crosslinks that occur just over time from the time we're made until, if we're lucky enough to make it to 100 years old, it just accumulates that damage. This damage is not reversible by enzymes, and not by drugs. And I'll get into that a little bit more in a bit. But it's fairly complex damage. And this is an example just for proteins. There's equally complex damage for the other macromolecules. These are covalent changes to proteins that are very difficult to undo, and in some cases are undoable. And my favorite example is a protein that is in our blood vessels. It's a major component of all our blood vessel walls, called elastin. And elastin undergoes all these forms of damage. And if we don't address that, that sets a limit to our human lifespan itself. And it is only one of hundreds of proteins that accumulate, altogether, hundreds of forms of damage to themselves, so over time. It's a very disheartening look at what happens to us as we get old. So as I mentioned, this is not just a protein, it happens to DNA, lipids, and carbohydrates. And we know that these forms of damage, at least for DNA and the extracellular matrix, are sufficient to cause all the other hallmarks of aging that people are focused on. So they're, in a way, looking at the effects of aging and not the cause, which is these forms of damage. So any strategy to slow, stop, or reverse aging needs to take into consideration this damage that accumulates. And so there are a minority of people in the longevity field that realize this, and their approach has largely been to try and target those individual forms of damage, like glycation is a popular one. If we could just reverse glycation, that would be a good first step, and then we can go down the road and try and reverse all the other forms of damage. For several reasons, I don't think that'll work. We can discuss it in more depth. The alternative is to just replace tissues, in which case you replace all the damage at once. You replace an old tissue with a young tissue that doesn't have any damage. So, tissue replacement makes sense for that reason. You replace all the damage at once, resetting the clock, at least for that tissue, back to zero. Some forms of damage include loss of information content, and those cannot be enzymatically repaired, or even less so with drugs. And then finding combinations of novel enzymatic drugs or other forms of molecular therapy that can target these often inaccessible extracellular forms of damage is highly unlikely without causing much worse side effects than doing good. So the approach that I think will eventually beat aging is a replacement approach. So in terms of implementability, replacements have been done for every part of the body except the brain already. So surgically, replacements are doable. Whether with biological replacements or with synthetic ones. So we know of artificial hearts. There are other parts of the body, like limbs especially, that are becoming more performant than biological ones. So replacements are well underway for every part of the body. We recently in the news, a man received a new heart that was developed in a pig. and in such a way that it would not easily get rejected by the patient. And there's a lot of efforts like this. So regenerative medicine is making great strides towards being able to replace every part of the body. The brain is a little more challenging, though. And I'll refer to the brain, but I'll be talking mostly about the neocortex, which is this outer folded part of the brain that is the largest part of the brain, and it's what defines us most as individuals. It's the conscious part of our brain. It houses our self-identity, and so that's the most important. So we're focusing on that first. All the other parts of the brain are essential to replace too if we're going to succeed in reversing aging. But for today, I'm just going to focus on the neocortex. And there are two reasons why neocortex tissue replacement makes sense. And one is the nature of how the neocortex works. It's incredibly plastic. So even during our lives, functions move from one substrate to another under certain conditions. This has been shown, and I'll give you an example of that. So our self-identity, our functions can move from one substrate to another if done slowly over time. So in principle, that means that we could progressively replace the entire brain. And also, the other reason is more recent work on the transplantation of very immature precursor cells that seem to ignore the fact that they're in the adult brain and do what they would if they were, for example, in a fetus or a newborn, and develop and make the appropriate connections with other parts of the brain and become functional. So that technically, or in practice, means that we should be able to progressively replace brain tissue as well. So I'll just give an example, as I mentioned, about plasticity. This is my favorite example, but there are others. So there are individuals of fairly advanced age who develop benign brain tumors. Sometimes they occur in the language center, and over the course of a few years, they'll completely eat away at that language center and destroy it. And eventually they need to go to the hospital because they have a really persistent bad headache or even a seizure because of the pressure that builds. And it's, bad news, good news. Bad news, you have brain tumor. Good news, it's operable. And so they surgically resect it with surrounding tissue for good measure. And those patients have been found to still be able to speak and never had an interruption in their ability to speak. And this is because if you give it enough time, the way that the neocortex functions, its plasticity allows, if you keep using those functions, allows those functions to move from one substrate to another. And there are many examples of this in the literature, in mammals that are used as model systems as well. So it's a really general property, and it's very encouraging from a regenerative or tissue replacement point of view. And the other reason I mentioned too is that these young precursor cells can develop the way they would in an infant and connect to the rest of the brain, right? So we don't have to understand the whole process of how the brain develops because these precursor cells know how to do it themselves. Nobody generated our brains, right? It's this developmental program that occurred, this innate program. And so what we're doing, our plan, is to just reestablish that program at a later time and in a different place in the adult brain. And so this is these are images from my lab, but other labs have reproduced this. It's not that uncommon now, actually. So we make these canonical lesions in the brain, and then we can rebuild the missing tissue. This is in a mouse, and this tissue develops very nicely in terms of its neuronal differentiation. exuberant projections to normal targets along normal axon fiber tracts. And so this allows us to perfect the tissue engineering aspect of what we're doing. And in addition to neuronal cells, we get vasculature developing in our grafts that fuses with host vasculature and circulates blood. So this allows us to expand the size of the tissue. And also vasculature, of course, is not only provides nutrients, but it also helps the development and the maturation of tissues. So this is an important aspect. And I like these movies. This is actually one of our grafts in the white here. It's a human graft in a mouse tissue. So we're engineering human tissue in mice because it's more convenient at first. And you're just looking through this cylindrical lesion that we've now recreated a neocortical tissue in. And you can see lots of projections coming off that. Here's just a blow up of one of those projections from a neuron. You can see a nice, on the right here, a nice growth cone for those of you who are familiar with neuroscience. That's the tip of a growing projection from a neuron when it's looking for its normal target. And we also have the formation of these very nice blood vessels in our graft. Again, these are human vessels in our human tissue that is in which in a adult mouse brain. So we're just using the mouse as a platform here to build our human tissue. So very nicely developed vessels. And we can visualize circulation through the vessels within grafts by labeling a small fraction of red blood cells with green dye, as you see here. So that's all good. Sounds very promising, right? But we're a very long way from having functional tissue. And that's because of the complexity of the brain, the number of cell types, and their connectivity. So, there are two ways in which we're testing this concept of progressive brain replacement. One is to engineer the tissue from scratch, and that's a lot of work. There are many cell types. What I've shown you in the last few slides are basically the principal neuronal cell type and the principal vascular cell type, vascular endothelial cells. But there are, and this is a gross simplification, there are some of the other cell types that we need to put together. There are more than this, but we have protocols for obtaining all of them. Even, we have about 50 cell types in the neocortex, and we have protocols for generating just about all of them. But then to reassemble them, they have to be in the right ratios, at the right relative maturity states, and we have to reassemble them in the right position, the right cytoarchitecture, embedded in, at least initially, a extracellular matrix that they are happy in and recognize as a developing fetal brain. This is our goal, is this mid-gestation human fetal neocortical tissue. That's what we want to engineer. Looks pretty simple with this stain. You just a multi-layered structure. But again, it's going to be a lot of work to get it to assemble properly. And then there's the then there's the post validation of function, which requires a lot of steps as well. So the quicker way of testing our concept is to use human fetal neocortical tissue. And so this is an example of a human fetal brain at mid-gestation, gestational week 19. And so we can take tissue directly from this, and it already has all the cell types in the right ratios at the relative differentiation states that they're supposed to be, and in the right organization. So we can directly transplant that as a shortcut in testing our approach. So we use both of those. And there's pros and cons to both. Like if we did want to use human fetal tissue in the clinic, which has been used for the brain before, we could do that, but the pros of the engineered tissue is scalability. And once we figured it out, scalability and speed, and custom in vivo shaping. The disadvantages, though, it's going to take a lot longer to develop, which means a higher cost. And we have to do this repeatedly for every part of the brain. Again, I was just focusing on the neocortex. The cloned fetal tissue has the big advantage of it's the real thing. So the functional aspect of it is already validated. It has shared costs with cloning if we use cloning for replacing other body parts and with other parts of the brain. So if we are to clone a brain, then a fetal brain, then we can use other parts of that tissue as well for replacement purposes. The disadvantage is scaling is very difficult, and surgically it's a little more challenging because you can't fill a lesion that you've created with the tissue. You have to shape the lesion. You have to match exactly the graft and the lesion you've created to put it in. But in any case, we're not going to do this directly for reversing aging. We're going to test this first clinically in a stroke model. So that's just illustrated here. I won't go through each step. But more importantly, eventually what we want to do is to be able to reverse aging. And this is this strategy I mentioned where we would do it progressively. So a certain percentage of the brain or the neocortex would be replaced every 3 years, like 15%. So the whole process would take, a decade or two to get full neocortical brain replacement in a fully rejuvenated brain. And this is, again, a very difficult project. And we have a large collaborative team. It actually needs to be a lot larger than this if we're going to accelerate progress. But we're off to a good start. And so, we just need to do more faster. And so if we succeed in this progressive brain replacement, replacing the rest of the body, I think is happening, more and more already. We'll be able to actually beat aging. So be interested to hear your thoughts on this and get any feedback or criticism. So I'll stop sharing.
Many thanks, Sean. That was fascinating. I'm sure there's going to be lots of deeper diving into some of these areas. Audience members are welcome to type questions into the Q&A window. Those of you watching along on YouTube, I can keep my eye on the comments window there and may feed points in from time to time as well. Sean, do all brains age? Isn't it possible that there are some organisms that are negligibly senescent, that they seem to keep their mortality rate constant regardless of how much time passes? And if that's the case, couldn't we learn from what's happening in these creatures' brains and try and copy it rather than having to replace brains?
Yeah, there are organisms that show negligible senescence, and that, in a way that I believe, so that they don't show an increase in pathology in their tissues, which I think is an important aspect that's often neglected today when looking at aging and rejuvenation effects. So there are organisms like that. They tend to have a very different mode of life than us, in the sense that I think all of them are continuously growing throughout life, which has two potential advantages, although this has not been proven as to why they show negligible senescence. And this is true for the brain and the rest of their bodies, is that with growth comes the opportunity to select against the damage, at least at the cellular level, and also to dilute any type of damage that's occurring. And so maybe that's the key for them. Of course, they could have other sort of genomic mechanisms of better repair, or of producing, for example, extracellular proteins that are more resistant to these forms of stochastic damage that I mentioned. So I guess the short answer is we don't know, but those are possibilities for why, but they would be difficult to implement in humans. Genome re-engineering of humans is an idea that I think is a good idea, but it's not going to help those of us around today. So, maybe down the road. And having humans be continuously growing is also probably not something that we want or we can imagine happening.
Just to stick with this one more round before I look at the audience questions, which are growing in number the whole time. And by the way, if you're in the audience, you can vote for each other's questions, which will make it easier for me to know which ones we should look at first. Just on the species that are negligibly senescent, and I can accept that lobsters or whales probably do get larger and larger over time, so there could be a dilution of any damage. What about naked mole rats? They don't seem to get much larger. Have you had a chance to look at them, or are they just too strange?
Yeah, I don't, I haven't looked. I know they're a popular model system for aging, or at least, an example of what we should be looking at. They are, I don't know that they're that strange, but I don't know to what extent damage of accumulation at the macromolecular level has been examined within them. And DNA damage is an easy one that people look at, but it's, by far not the only form. So whether they are truly negligibly senescent, I don't know for sure myself. It may be known. And if so, then it would be the sort of the latter possibility that they have just evolved a genome that is much better at minimizing accumulation of damage, or repairing. Some forms are repairable, so maybe repairing damage, like DNA damage is repairable. A lot of the extracellular damage is not. So yeah, I don't know. But, it's interesting. It would be interesting to find out why. But again, how do we translate that to humans? Do we want to re-engineer the human genome? If we do, good for our progeny.
Peter Morgan asks questions about memories. He asks, what happens to our memories as we slowly replace parts of the brain over time? Do our memories get kept intact, or might we lose memories that we'd forgotten we'd had? Yeah,
so in order to keep memories, and this is true without replacements, you need to occasionally recall them, or else they will disappear. That happens to us all the time. We forget things over time, and the more we think about things, the less we forget them. So that's sort of part of the principle of plasticity. It's very, very use-based. Our brains have evolved to be useful to us. There's things we don't think about. We tend to forget them because it's not useful. So the same applies for the replacement strategy. As long as, so I kind of glossed over the protocol there, so I'm kind of glad you're asking this. So along with the progressive replacement, what we do is progressive silencing of identified functional areas, like the benign brain tumor was doing, except we're not going to use tumors to do that. But if it's a progressive silencing of a function at the same time as you're still using that function and providing a new substrate, you won't lose it. You won't lose those memories, you won't lose those functions, and you won't lose, your self-identity. So as long as you, make a point of recalling those memories, and this is standard practice in brain surgery, is before you cut anything, you go in there with an electrode and map what those functions are, because oftentimes there are things you want to avoid with the knife. So you'll know, you'll have some idea what's there, and you'll just want to make a point of remembering that or recalling that over the 2-3 year process when you change the substrate for that area of the brain. That's the idea.
How do you identify the 15% of the brain that you would want to change in any sequence? How do you know which is the oldest parts, or is it not done like that?
Yeah, no, that 15% is, sort of a guesstimate of what, again, based on examples that happen in real life of, how much plasticity we can afford to engage without, reverting to a too immature stage, right? And to some extent, it will be done based on functions. So, there's modular functions like motor functions, visual functions, and so we're going to probably want to replace those at least unilaterally at the same time. So that contributes to sort of in part to the 15%, but it also depends on how much we're willing to risk in terms of forgetting certain things. So if we replace bigger and bigger areas at once, then yeah, we're going to have this great new plastic replacement area that might make it easier for us to learn a new language, but at the same time we might be more at risk of forgetting things because the more functions, more memories are encoded in larger areas, and so to sort of ensure that none of that is lost and being used all the time might be a little more challenging. But there may be some level of personal preference here. So for some people, maybe doing half the brain at once because they're old, they don't have that much time. They might prefer to do it that way than just 15% every 3 years or so. Or if you start young, maybe and the surgical techniques are perfected, then maybe you'll want to do less than that. But this has to be tested more empirically, obviously, and to determine what exactly would be the ideal rate of replacement.
You offered two methods, A and B, for generating new parts of brains. One is to grow them almost from scratch, and another one is to use fetal stems, parts of fetal brains. Tony Zarneski asks, what about 3D bioprinting? Does that cover what you talked about before, or is there something alternative in 3D bioprinting that could be used?
Yeah, so 3D bioprinting would be along the lines of engineering the tissue from scratch, because you would still need to print the cell types in the right ratios and the right positions, but you would do it with a printer instead of manually, which is what we do in the lab now. I have a student who's very talented at that. But yeah, so 3D bioprinting would definitely be an advantage, in the future to be able to do this in a more automated way. Our tissue reconstruction is done in vivo, so we do not make the tissue and then transplant it for reasons of connectivity. So you could still do in vivo 3D printing. It's been done before. It's not very common though, so it would need to be perfected. And it would also, mean that eventually, with smart robotic surgeons, you could do this more effectively and more routinely in people. So I would definitely like to see advances made in vivo 3D printing and smart robotic surgeons, which those things are already happening, but and hopefully by the time we're ready to implement this in humans, it will be a reality, so we can take advantage of it.
So tell me more about these smart robotic surgeons, P&J. comments that deploying surgical changes at scale would currently be expensive and unlikely to scale to whole populations. Can you imagine what mechanisms would make this easier to deploy?
Yeah, so again, automation, like I just mentioned, might help a little bit. The production of the cells will probably, remain expensive compared to, drug manufacturing. So yeah, it's a decision that we'd have to make. Is do we want to continue aging, getting sick, and dying, or do we want to invest a little more in this as a society and cover the costs? And in all honesty, others have done the math on this, and there's a tremendous amount of savings in keeping us young, healthy, and employed, as opposed to sick, unemployed, and a burden on society, at least financially.
So this is the general argument of the longevity dividend, that in principle, if we could find suitable treatments, they would have tremendous economic benefits because people wouldn't be sick, people wouldn't be consuming healthcare resources, there would be active members of the economy, there would be producers and consumers. But that does depend on us finding relatively cheap therapies. And the question comes back, it does seem like an incredibly complicated operation. It might cost hundreds of thousands of pounds. Do you think that with sufficient research, that costs could come down via the automation that you're considering?
Yeah, I think so. I mean, the materials are not expensive. It's the labor, it's the work that's being put in. So if things are automated, the cost should come down. I mean, I haven't looked deeply into that, but I would expect that would be the case. There are companies now that are automating the process of taking your fibroblast cells and turning them into these induced pluripotent stem cells that can then give rise to every part of the body. So there's fully automated protocols for that. So things like that should bring down the costs considerably, because again, the materials you put in are initially are not expensive.
A question about these induced pluripotent stem cells. So they come from parts of the body already, so maybe from a skin graft. But that skin is already old, isn't it? It's got macromolecular damage in it. And the process to turn it into a stem cell, the Yamanaka factors, that can give it its pluripotency, but are we sure that it actually undoes sufficient of the macromolecular damage in the cell so that it is good to deploy? Or would it actually be bringing old materials into the body as well?
Yeah, this actually reminds me of your first question about why there are some organisms that do not show senescence, and the reason is pretty clear, I guess, for this reprogramming of fibroblasts to iPS cells is that there's a tremendous amount of cell proliferation that goes on between, a fibroblast being reprogrammed and obtaining an iPS cell at the end, and a tremendous amount of selection. The efficiency is less than 1%. So you're getting a lot of selection and dilution of any type of damage. Plus, you're not dealing with any extracellular matrix, which is a major problem when you're talking about a whole organism. But when you're talking about cells in culture, it doesn't exist, really. It's pretty clear why you get rid of damage in that case. And you can also do very good quality control, even at the DNA, transcriptomic, and protein levels, to make sure you really have a young cell. It's actually this sort of very similar to the process of reproduction from generation to generation. We have germ cells in our body that some of them may accumulate damage, and they don't get passed on, but there's a selection. There's a lot of selection and growth that goes on in transmitting, creating a new person for the next generation. So that's sort of a similar process there as well.
There are a couple of suggested alternative mechanisms. Brian Collins, who's watching on YouTube's, wonders, why can't we just stimulate partial reprogramming of the brain by reactivating some of the dormant or quiescent stem cells or progenitor brain cells that's in the brain already? Might that be less intrusive and a more successful way to get new cells into the brain?
So there's a lot of misconceptions there. Those progenitors, this adult neurogenesis that occurs, first of all, it's still a little bit controversial whether it occurs at all in humans. Let's say it does. It's in a very restricted part of the brain. It's the dentate gyrus of the hippocampus, which is minuscule compared to the neocortex. Not only that, but those stem cells are, restricted in the type of neuron they give rise to, right, which is appropriate for that part of the brain, but not appropriate for other parts of the brain. So, yeah, you could stimulate these to give rise to more neurons, but it's really not going to do much in terms of, addressing the damage in the rest of the brain. The idea of reprogramming too is I think it will have some uses in potentially helping tissues regenerate better in the adult, but it's still going to be very difficult to implement compared with just putting in already immature cells. So the reprogram or partial reprogramming with Yamanaka factors, what that process does is de-differentiate cells. And we already know how to make those cells. We could already put them in. But the process of partially de-differentiating them in vivo, implementing that is going to be much more difficult and problematic. For those applications where we just want to replace cells and not tissues, which again will not address all the problems or even the more important problems of aging, there's already an easier route, more direct route to doing that than trying to reprogram cells in vivo.
And when you say easier route, the one that you're exploring, how far away with a favorable wind is it before this could be deployed in real humans? Is this something that could happen within a decade or two, or?
It's already happening for different parts of the body, including for the brain, right? We've been doing this for Parkinson's for decades. We haven't been trying to reprogram cells to de-differentiate and give rise to new substantia nigra neurons. We just transplant them in, and in some cases, it's not fully perfected yet, but in some cases, patients have been essentially cured. Some have gotten worse, which is why to reassess how it's being done. But some have been essentially cured. So we know in principle it's possible. Compare that to what it would take just to generate those new neurons by cellular reprogramming. It's just decades behind.
So these treatments for various brain disorders, I think I remember Professor Stephen Minger giving a talk to London Futurists a few years back, referring to work done in Sweden several decades ago, in which some of the transplanted cells seem to be successful 20 or 30 years later in the individuals concerned.
Right. That's right. Yeah. And those were taken from human fetuses, but they did remarkably well. So what people are doing now is, again, trying to derive them from these human pluripotent stem cells so that they can scale it and use it much more broadly and not be reliant on fetal tissue, which is a more variable source, which is why, although some people did remarkably well for decades after the transplant, they could go off the L-DOPA, their symptoms disappeared. Others got worse because of the imprecision of getting the right cell type from fetuses. And so that's why we're more hopeful that there'll be consistent results using much more controlled cells derived from human pluripotent stem cell sources.
That brings me to a question asked by Christian Fried, who says they're not in this camp themselves, but isn't there criticism related to messing with natural processes of aging? And any mention of using fetal brain cells probably stimulates quite a lot of yuck reactions. Even if we don't do that, even if we use the iPS cells instead. What's your response to that reaction?
I mean, that's what medicine is. That's what we've been doing in medicine forever. With, you know, we've been doing organ transplants. At first, I was like, oh my God, that is so unnatural. And now it's routine. There's 100,000, or heart transplants done, I think, just in the U.S. It's just very routine now. I think it's just anything new like this is going to be, make some people, think that it's bad because it's different or it's new. But I mean, if what we're doing is talking about maintaining health, maintaining people's lives for longer, I think it's hard to argue that that is a good thing. It will always be somebody's choice. If they don't want these treatments, that's fine. But for those who want their loved ones to live longer and themselves to live longer, healthier, yeah.
Another alternative suggestion is that some organs could be replaced not by biology, but by synthetic equivalents. So there are examples of people with synthetic hearts, apparently. I don't know how common this is. Some of the other organs might be done differently to how nature creates them. Is there any suggestion that part of the brain could be replaced by synthetic equivalents instead, or is it entirely a straightforward biological replacement that you've been exploring?
Well, yeah, we've only been exploring the biological replacement, but in principle, it doesn't have to be biological. It could be synthetic or silicon-based or whatever. The important thing is the continuity of function, the continuity of self. It's, the idea, for example, of just downloading a brain or uploading your mind is just really what it is creating a copy. It's not preserving who you are, right? And that's pretty easy to demonstrate, at least in a thought experiment. But the issue is, so for progressive synthetic replacement of the brain, which eventually, down the road, I hope we get to, because it's probably easier to deal with than biology, which is very complicated and designed by millions of years of evolution instead of rationally designed by, a good engineer. We're just nowhere near that. I mean, having that technology that can sort of a synthetic tissue that can mimic what the brain does. I mean, just think of deep learning neural nets now. You have to provide them with hundreds or thousands of examples of what a cat is for them to learn that. Think of a child, they see a cat once and they know what a cat is. You can show them a purple cat, a big cat, a small cat, a well-lit cat, a dimly lit cat. They go, a cat, they learn instantly. There's something about how the brain functions and how we learn that, we're just nowhere near reproducing in the synthetic world yet, but hopefully down the road, I guess.
I think you've touched on the question that Gordon Silverman has asked, who talked about creating a complete brain somehow, but without having, I guess, a consciousness attached to it, and then using that as a means to transfer all your existing memories, thought processes, patterns of consciousness into. What's your comment on that proposal?
Yeah, I mean, it is sort of what I was just talking about. As long as you could do that in with some form of continuity, um, then maybe that would be a good idea at some point if you could do that. But yeah, again, there's no really technological basis for thinking we can do that yet.
But equally, there's a long way to go with your own proposals. As you've said, there are creation of the right kind of cell types. There's figuring out the connectivity between the cell types. There's getting them at the right stage of maturity. There's embedding them in an extracellular matrix. And there's removing, presumably, the older cells in order to make room for these new ones. That seems like still a very ambitious project.
Yeah,
Again, with a favorable wind, what timescales do you think before you could say, yeah, this is essentially mastered?
not before 5 years, and that's with a real change of pace here. I mean, all those points you mentioned are challenges that we need to achieve, but the technologies exist. That we're not inventing new technologies to make this happen. We just have to empirically determine what is working best. We can derive these different cell types already, and so we know how to test them. We can do high resolution quality control. So we just haven't done it, and so we need to put all those things together. So 5 years is optimistic, but not impossible. At the pace we're going now, without much acceleration, 50 years would be optimistic, I think.
Did you say 50 years?
At the pace we're going now, yeah, 50. So, for this to work in some of our lifetimes, yeah, we definitely need to accelerate progress. But we don't have to invent new technologies, right? It's very much like the idea of putting someone on the moon in the early 60s. We knew it was possible. We just didn't work out the details yet. And for someone to say, oh, by the end of the decade, let's do it. It was like, okay, well, now we have to work out the details, and we did it. So as implausible as it seems, it's just a matter of working out the details.
So when Kennedy made that announcement, there were various rocket engineers and so on who could evaluate it, and they could say, well, yes, it is a challenge, but here are some ideas. What's the response when you make these suggestions? What's the response from your fellow brain scientists or people in the anti-aging field generally? Are they saying, yeah, this sounds interesting, we should put more research into it? Or are factors holding them back from adopting a similar line of research themselves?
It's been remarkably positive. And the only thing holding back people from more actively working on this in this field is the scale of it. Again, if we want to make the comparison to the Apollo project, if you ask these engineers to just work in their own little labs independently and come up with a solution to putting someone on the moon, that would have taken forever, if at all it would have happened. It's only when they all come together and work, and that's what we're missing here. As I mentioned, we have a group of collaborators that are addressing different aspects that we need to address in order to get this to work. But that's the challenging thing in the current, funding mechanisms, government funding mechanisms. They fund little labs independently. There are no large projects that are willing to say, yeah, we want to come up with a technology where we can repair or reverse age-related damage in the brain. No one has stepped forth and put up the money necessary to bring these scientists to work together to make it happen. But the response I've gotten from people, neuroscientists in the field and stem cell biologists, is almost unanimously excitement. So
so What's holding up that excitement from turning into people adopting the same research? I mean, if I look at the drug approach to anti-aging, there's a big pharma lurking in the background, and big pharma potentially can make lots of money out of suitable, some of the drugs at least. Are there similar commercial forces that could be unleashed in support of the work that you're doing, or is it just academic and government money that you could hope to reach?
Yeah, I don't know. I think it is from what I'm experiencing now, going after much larger sources of funds to move this along much faster. It is the private sector industry that is taking the first steps forward to back this kind of Apollo type project. And so I'm hopeful that that we'll get it done. And hopefully, the governments will follow suit, say, okay, look, this is for real, this can happen. Let's put together an effort to do so as well, or to join the effort. The sequencing of the human genome was an example of that, right? There was a private effort, and then there was a government effort, and in the end, they ended up collaborating.
Craig Venter and Francis Collins, the two different groups, had different backing to start off with, but in the end they could benefit from the work each other were doing.
Yeah.
So in the spirit of trying to find less expensive ways to do some of this, Brian Collins in YouTube has suggested, could there be ways to deliver things via nasal cavity, blood vessels, or using, I don't understand all of this, electro or ultrasound poration to make the blood-brain barrier more accessible to stem cells? So less intrusive, perhaps, than drilling holes in the skull.
Yeah, no, I think the short answer is no, because it's not just cells. If it were just cells, then yes. But to reverse aging, we have to replace the extracellular matrix. We have to replace the blood vessels in tissues and all the other cell types. And that, I can't imagine a way of doing that through, the nasal passages that have been used for other things, as well as circulation. Even with guided electromagnetic beads, which some people have used, are developing as well, which is a very neat technique. But building a tissue with them, I think, yeah, that would be very difficult. It's not impossible, without, again, having more bad side effects than benefits.
Another alternative approach is referenced by Didier Cornell. Now, you may not have read this, but he says there was gene therapy injections of a group of people in Mexico via BioViva Sciences, which injected something that stimulated telomerase, which made the telomeres longer. And somehow it looks as if the patients who received this, they are performing better on cognitive tests than before. And it presumably isn't because the telomeres allow neurogenesis, because in the brain there isn't much stimulation of new neurons. Have you had a look at any of that research that gene therapy could be involved in improving the brain functioning?
Yeah, I haven't followed that example. I mean, gene therapy is a very effective way at treating certain diseases, especially ones where the underlying cause is a genetic lesion. So it's particularly well suited for those. Yeah, there's another point that's brought up here. So there are good uses for gene therapy, even in replacement strategies, right? If we're replacing somebody who's at high risk of stroke or something, or Alzheimer's, and there's a known genetic determinant in their family, it would be fairly easy to gene correct that before making the tissue from that patient's cells in order to avoid that problem in the future. So, you know, gene therapy is great in that sense. But, you know, we have to be very prudent, I think, in equating improvements in cognitive function to rejuvenation. I take caffeine, you know, fairly regularly, and my cognitive function improves tremendously. Have I just been rejuvenated? In a sense, if that's what you want to call rejuvenation. But, you know, in terms of my rate of damage accumulation in the brain, I don't think it's effective. So there's a lot of things we can do that sort of, you know, increase performance, but don't necessarily address aging. So we have to be very careful of what we're looking at in terms of markers of aging.
This is something that quite struck me as I was reading your book, that there may be many treatments that could reduce the incidence of various diseases that we normally call age-related diseases, but still there is the ongoing accumulation of macro damage to the macromolecules, so that people will still likely then die at 115 to 120 at maximum, regardless of these other therapies.
Yeah, exactly. Yeah. You know, a 100-year-old person, there are some out there that are essentially disease-free, you know. But again, they're not a 20-year-old, right? I mean, this is pretty obvious. They still aged, even in the absence of disease. And so for some people in the longevity field, that's the goal, is just to get us all to that age, you know, 100, 110, and that'd be great. And I think that's great too. I mean, you know, the longer we live, the better, and the healthier we are, the better. But I think, you know, we can go beyond that. We can defeat aging and live much longer. But that will take different approaches, like the ones I'm suggesting.
Let's talk about possible game changers in the sense of something that would make the public or other researchers suddenly sit up and say, wow, this is something I didn't really expect. A bit like, I don't know, in the world of AI, there's AlphaFold, in which software is able to predict how proteins will fold. And more people are saying, well, this means that AI has gone beyond simple software regression into something real. So what could be an experimental breakthrough in your field that would make more researchers want to give attention and could unlock more funding?
Yeah, I think for the brain, it would be just a proof of concept that you could replace a part of the neocortex. You know, so you could design an experiment where if you remove part of the cortex, you lose that function, but if you replace it, you can reacquire the function, and you can prove that it's the neuronal activity, the electrophysiological activity of that new tissue that is underlying the new reacquired useful behavior to that animal, because it's not likely to occur in humans first. I think that would be, you know, a major turning point, because it validates the concept.
A little bit like what Ted Berger from San Diego did a decade or so ago, when he showed that some parts of the hippocampus could be damaged and then replaced by, in this case, an electronic link. And that showed people that his line of research at least had more promise. So potentially there could be rats that are trained to do some task, and that if their brains are augmented in this way, those who just have the brain part removed lose the ability to do the task, but those who've had it replaced gain it. So proof of concept.
Yeah.
Can we talk about head transplants as another possible game changer? Because I mean, when I was looking at this, 6 or 7 years ago. It was almost on the point of somebody doing a head transplant. Some poor person had a body that was very severely damaged and he thought he was going to die, and he was willing to pay for his head to be transplanted onto another body. That isn't quite the same, but it would involve rewiring large parts of the nervous system. Is that likely to happen anytime soon, do you think?
I don't know about the, so here's my take on it. The quickest way to beat aging would be through whole body replacement. Right? You replace everything at once. You could, so we've cloned primates before, right? That was done. We've done head transplants in primates. Robert White did that in the 70s successfully, despite rejection that occurs pretty quickly if they're not immune matched. And we can bypass the spinal cord. So this neuronal rewiring would be nice if it works, but there are other means that have already been validated. So there's quadriplegics with brain-machine interfaces that were able to use their arms again. Elon Musk has this company Neuralink that is making this, repeating this in a much more practical way where people can actually walk around with the device, whereas previously, those quadriplegics only regained function of their arm when they were in the lab with, all the computation power needed for them to be able to do it. So really couldn't take it home, so it wasn't of much use, but that will be overcome. So we know how to clone bodies. We know how to do head transplants. We know how to bypass the severing of the spinal cord. Yeah, sure, they're not ready for humans yet. We need to perfect that a little, but it's not science fiction. Those technologies are, are there. Again, with need for some improvements in efficiency and consistency, but that's totally doable. So that would be the quickest way, right, to completely rejuvenate your body, is to get a new young body, a new you. And then, of course, then we still have to deal with the brain. And so we still need to do this develop this progressive brain tissue replacement to do that. But my take on head transplants is if we're really in a hurry to defeat aging, that's part of the equation.
Now, something that's possibly more accessible more quickly is what you mentioned with stroke recuperation. You had a slide that actually you went past quite quickly. I'd be quite interested if you brought it up again, if that's possible, because that's stroke is something that affects people all the time, and if mechanisms could be used to help people recover from strokes, I think that's going to make the world sit up and listen.
Yeah, so this is just a cartoon of what we would do, and it is again, I'm mentioning that I'm getting industry interested in this, in funding this, and of course for that there need to be intermediate milestones, and stroke would be the first intermediate milestone in the clinic that we would target, more than likely. Again, because it's so common, right, as you mentioned, and it would be very significant in terms of people's health. Here, there is no, unlike the slow-growing benign tumor that allows the plasticity to move a function, a stroke is a catastrophic event where the function that's lost, motor control, or language, or vision, if it happens in the occipital cortex, can be lost forever. So our goal in this case is to provide this sort of tabula rasa new tissue with tremendous plasticity, the equivalent of a one-year-old fetal neocortical tissue, so that the individuals would be able to relearn whatever ability they've lost with relative ease. And so this, again, is just a depiction of that same process where we would take patient. It doesn't have to come from the patient. It could come from generic allogenic human embryonic stem cells. A lot of the current brain cell transplants being done in humans come from ES cells. Most of them do, but ideally it would be patient derived. And So you wouldn't have to use immunosuppressants, but also you would basically, if your genetics dictates to some extent, who you are as an individual in terms of your brain function, then you would like to preserve that as much as possible, which is why this would be preferable to use a patient's own cells, and then generate the different cell types and reassemble rebuild the tissue at the site of the infarct.
And how long does that process take? If the person has the stroke, you take the fibroblasts, you take a skin biopsy. How many months is involved in growing sufficient cells to apply them?
Yeah, so we could do it in a reasonable amount of time because in a lot of cases, right, in the real world, we're going to target patients that are not showing any recovery, which is a fair amount of like half of stroke patients are permanently disabled, those that survive. So there's a third that die from the stroke, a third that can recover sufficient function to go on with their lives, and a third that remain permanently disabled. Those are our targets, but you can't always tell them right away. And so you need to wait a few months anyway. So there's a window of time there that we can, where we can do this. I mean, we can generate these cell types within a couple months. So
that's, that would be feasible. That's not too long to wait.
Feasible. But again, if we're using allogenic cells and we have a bank of these cell types already, we could do it much faster. So it depends.
The cost of coping with possible rejection. You have to match in some sense. You'd find
patients would be on immune suppressants for the rest of their lives, like everybody who receives an organ transplant or allogenic cell transplant. Correct. Yeah. But in terms of scaling and commercialization, it is right now a more attractive model.
So we're almost out of time. I will ask the audience if there are final questions anybody wants to add into the mix as closing thoughts. One question we haven't come to yet is by Gordon Silverman, who just asked, how does the damage progress through the brain? Is the deterioration linear, that is one section to an adjacent section, or does it tend to be disjoint happening all over the brain?
Yeah, it does happen all over, and again, not enough work has been done to characterize the damage, although from my perspective, enough to know that it's going to be impossible to deal with molecular treatments. But some forms of damage occur linearly over our lifetimes, like racemization. It's sort of just a physical process. And there are others that may accelerate with the hardening of the blood vessels, for example, which is not a good thing. And so that may have accelerating effects on other forms of damage. But, short answer is I don't really know, but it does occur in all parts of the brain.
So then it comes back to the question I asked you earlier, how do you decide which of the 15%, which 15% of the brain do you replace at any one time? And that's an engineering question, I guess.
Yeah. I mean, that still needs to be worked out. I mean, that's why we're talking to systems neuroscientists, who know how the different pieces fit together and how we might be able to modularly replace things in terms of functions without, disruption or interruption in the ability to do things. So, 15% is sort of a guesstimate at this point, but it needs to be worked out.
And other parts of the world that are more responsive to your ideas, that when you talk, are there cultures, are there locations that people say, oh no, we don't want this at all, and others are saying, well, actually, we would be very interested if there's only some funding available?
Yeah, again, maybe it's the circles I hang out in, but it's been generally well received. I do talk to people that say, aging, just aging itself is like, no, we shouldn't get rid of it, even though it causes a lot of people to suffer for decades and then they die and we lose loved ones. But they say it gives meaning to life, for example, and without aging, life has less meaning. I don't know. You can always do high risk activities and be closer to death if that makes you feel more alive. People do that already, right? But there are some people who are just generally against the idea of not aging or reversing aging. But beyond that, the main objection is just that this involves surgery. Even though people get brain surgery, even multiple brain surgeries, routinely. It's still, for most of us, a scary idea to undergo surgical replacement approaches. So I think that's a lot of the sort of fear is that this seems very radical, even though brain surgeons are excited about, like we're working with a brain surgeon now who's excited about trying this, and thinks it's totally possible. So,
well, I can sympathize with people saying they'd much rather take some medicine than have their heads cut open.
of course, yeah. But the medicine is not going to work for aging, unfortunately.
How much of the skull would be cut in order to do these operations? If you're talking about changing 15% of the brain, it seems like a very intrusive operation.
Yeah, I mean, the way that brain surgery is done now is, you remove the, it's basically a skull flap that we then replace afterwards. And we do this in the lab all the time too with animals. Then you can easily do it, progressively as well. And I think it's actually a good opportunity to replace the skull with the younger skull as well, because all parts of the body are going to need to be replaced to be aging. So it's the skull, it's the brain, and it's the layers in between them as well. So there's meninges between the skull and the brain that all need to be replaced. And so, we could, down the road, consider doing this all at the same time.
So we'd replace the skull with something that's more easily openable in the future, so that it wouldn't be so traumatic. It would be designed for upgrades rather than designed to be closed.
You can imagine doing that as well. Yeah.
Interesting.
We haven't worked that out yet.
If people are listening to this, either live or on the recording, and if they say, well, this sounds like a really important idea, how could they help?
That's a good question. So what we need right now are funds. So if they're in a position to be able to do that, that'd be great. Second to that, what we need is people working on this. So they're well-trained people. We need computational biologists, all sorts of neuroscientists, stem cell biologists, and so we need more people working on these problems together in a concerted way to make it happen.
And part of the raising awareness is to point people at your book, to help draw attention to the book, to review it. You mentioned to me in another discussion it was almost 2 years old, some parts of the book now. Are you going to be updating it, or do you have a new book underway?
Yeah, it's a good thing to keep in mind if you do decide to read the book, because we've made progress since then. And I was optimistic then, and I'm much more optimistic now about regenerative medicine providing the answers that we need. But I do not have plans for a second edition, primarily because I wrote the book as sort of a business calling card to all these potential private sector investors, and just basically sent it out to them for free. And that sort of mission accomplished to some extent. So I made some connections there that are being actively pursued. So my point in writing the book was to raise awareness of this approach generally, but also from a very practical point of view to make things happen. And that seems to be underway. So yeah, I have no plans right now for another mission.
Understood. But you can keep doing videos, you can keep appearing on various shows to help spread the most latest up-to-date information.
Yeah, yeah. And that's another thing, you know, that in terms of helping is what you're doing and what others can do as well is just talk about this. You know, if we do want to beat aging, I mean, there's that, I don't know if people are aware, and if you know, David, of the fable of the dragon tyrant. You may have read that. Yeah. So, you know, that's a good sort of starter, I think, for anybody who, you know, thinks that aging is inevitable, or you know, how we'll go about eventually beating it.
And I think there's a good video to illustrate it these days as well. To turn to Nick Bostrom's prose from, I don't know, about 15 years ago into even more vivid communication.
Yeah.
I'll add a link in the video description. In a moment, I'll come back to you, Jean, in case there are final remarks that you think should be added in. Let me briefly just talk about the future of London Futurists. Today, this is what we've been looking at, this particular event. We're going to come back to the subject of aging in 5 weeks' time with a different angle. That's on Saturday, the 26th of February. A different speaker, a lecturer in philosophy in various colleges in and around New York. He has written a book called The Case Against Death, and he is unafraid to say that death is evil. So what we'll be doing in that event is less discussing science, but more discussing a question which I see William Young has just dropped into the Q&A, which is more philosophically, are there other arguments about the soundness or the morality of what we're doing here? Something we haven't really given time to in this meeting, but there'll be plenty of time in that meeting in 5 weeks' time. So that's the future of thinking about death. In between that, there's one more meeting that's been announced. If we think death is bad news, then death on the hands of superintelligence will be bad news as well. That meeting is featuring Professor Roman Yampolkowski, who is somebody who's given a great deal of thought as to how we could control artificial superintelligence when it might emerge. If we control it properly, it could help us to solve problems of aging much more quickly, whether by drugs or whether by the replacements, stem cell therapies, tissue engineering that we've been discussing today. But that assumes we can control it, and there are many arguments that say that we won't be able to control it. It will be just like gorillas, chimpanzees thinking they could control humans. So that's what we'll be looking at in 2 weeks' time from today. That brings us back to today's event, which is being a tour de force by Jean Hébert on a different angle in looking at aging. Jean, is there anything you'd like to add by way of closing thoughts?
No, but I'm seriously considered joining you for your next guest. I'm a big fan of Ingmar Patrick Linden. So yeah, so anybody who's you know, questioning whether or not aging is bad or not, I would recommend tuning in.
Well, the argument in the book, in essence, is that many philosophers suggest that it's the wise thing to do to embrace aging and death. This is what mature people do, and it's only short-sighted, adolescent, egotistical people who want to defeat aging. And he systematically refutes that. So he argues against the likes of Buddha and the Stoics and lots of Christian philosophers. So it's quite blistering stuff. He accepts it's a negative polemic, but he argues it's a very positive and liberating view. And if we believe that life is good, then we should accept the consequence that death is bad and do something about it, which would include getting more funds to the kinds of projects you are doing, Sean.
Yeah. I'm no philosopher, but I guess that is the perspective I have. If we value a human being, then why would we not want to stop the suffering and death of that individual?
So in a moment, I'm going to switch off the recording and have a short break. There will probably be just a small number of people gathering in an after session here for a little bit of chewing the cud, reflecting on some of the discussions. Anybody who is watching in the Zoom is welcome to hang around for that. Those who need to leave, I understand you may have other things to do in the rest of the Saturday. And it remains for me to say thank you very much, Jean, for sharing these ideas. I strongly recommend people dig into your book, which is accessible, nice short bite-sized chapters. Each has substantial content. And I think it will help people to think differently about solutions for aging. Thank you very much and goodbye.
Thanks for having me.